In this video, I'm going to talk about nucleic acids. This is the fourth major biological molecule that we've looked at. We've looked at carbohydrates, lipids, proteins, and now nucleic acids. And you could probably make the case that nucleic acids are the most important of those four, because nucleic acids provide the code for which to make the other ones. So you've got DNA, RNA, and ATP. Let's go back a slide there. Right, DNA, RNA, and ATP. So DNA provides the genetic code that distinguishes you from me and us from plants and all the other living things on Earth. And ATP, that's the energy currency in the body, as you know. So as you're writing your notes here and thinking and listening to this video, it's using energy, and that energy comes in the form of a molecule called ATP. So you know that sugar provides you with energy, but it's a little bit more complicated than that. The sugar is actually used in a reaction with oxygen called cellular respiration to make this molecule ATP, and that's what's used for energy. So we'll start with DNA and RNA. We've mentioned that proteins are coded for by genes. This is something you learned about a little bit in grade 9. And genes are found on the DNA. Well, if you took the DNA from one cell, it would stretch out about 2 meters, which is pretty amazing to think about. And in that 2 meters of DNA, you might be surprised to know that only about 30% of that is genes. So you're probably wondering what the other 70% does. Well, when I was in high school, we called it junk DNA, because we thought it was junk, just left over from years of evolution. But we now know that it's not junk, that it serves a purpose, and something we'll talk about a little bit more in class. So just like the other biological molecules, DNA and RNA are polymers, and they're made up of individual building blocks called monomers. In this case, the monomers are called nucleotides. So you could say if you put a whole bunch of these nucleotides together to make DNA or RNA, you could say that DNA and RNA are called polynucleotides. And one other interesting thing about DNA is that it's made up of 4 different nitrogenous bases, abbreviated C, G, A, and T. And it's interesting to know that all the living things on the planet share those 4 bases, and just in a different order, which is kind of neat. So the DNA, you'll also remember it's a double helix. So there's 2 strands twisted around each other. You've got a sugar and phosphate backbone represented in blue here, and then you've got these nitrogenous bases, and they base pair. We'll talk about that in a second. But first, just look at the difference in structure between DNA and RNA. RNA is a single strand, so no double helix. It's also a sugar phosphate backbone, and it's made up of C and G and A. And here's one of the differences. It doesn't have thymine, or T. It has uracil, abbreviated U. Okay. There's also a difference with the sugar in that sugar phosphate backbone. We'll talk about that in a minute. So looking at these bases a little bit closer, a couple things to point out. First of all, the C, the cytosine, and the T, the thymine, you'll notice they're made up of 1 ring of atoms, whereas the guanine and the adenine are double rings, a 6-sided ring with a 5-sided ring. And you might remember that the base on one side of the DNA strand forms a hydrogen bond with the base on the other end of the DNA strand, and the C's, the cytosines, always bond with G, the guanines, and the A, the adenines, always bond with T, the thymines. This is called complementary base pairing, and this is a hydrogen bond right there. And so if this DNA molecule is going to need to unwind for some reason, like to replicate itself, or to make an mRNA molecule which is going to go off and provide the code for a protein, if it's going to break, it's going to have to break these hydrogen bonds. And there's an enzyme that can do that, an enzyme called helicase, because it breaks the helix. We'll talk more about the process of going from DNA to a protein in the next unit. This is called protein synthesis. But right now we're just going to focus on the structure. How many genes are there in the human genome? Well, we figured this out not that long ago, just over 10 years ago. Scientists used to think it was probably hundreds of thousands because we're such complicated creatures. We were a little surprised to find that it's only about 23,000 genes. You know, those genes can interact with each other in many different ways, but it's only about 23,000 genes. Compare that with the number of genes in a black cottonwood tree. Now you might think there'd be a lot less because it's a tree, and surely we're more complex than a tree. But there's about 45,000 genes in a cottonwood tree. So the number of genes is not necessarily indicative of the complexity of the species. Okay, there's a lot of complexity in how these genes get turned on and off and how they interact with one another. But it's not just the number of genes. And each of these genes provides the code for a unique protein. So those monomers we talked about, the building blocks of DNA and RNA, called nucleotides, are made up of three parts again. The nitrogenous base, a 5-sided sugar, and because it's got 5 sides, it's called pentose, and a phosphate group. There's a simplified model. So here's the phosphate, and this is the sugar, the pentose, and that's going to go bond to another phosphate, which is going to go down and bond to another pentose. So that's the sugar phosphate backbone. Here it is in a bit more detail where it actually shows the atoms. This should say phosphate group. And this shows the complementary base pairing we mentioned before. Now remember how the cytosine and the thymine were made of one 6-sided ring? Okay, the same is true for uracil in the RNA. And these single-ringed nitrogenous bases are called pyrimidines. And the guanine and the adenine, which are single rings, are called purines. Okay, and you remember that the C's and the G's, complementary base pair, and the T's and the A's, or the U's and the A's if you're talking about RNA and DNA. So what we're saying here is a pyrimidine is always going to hydrogen bond with a purine. And in this diagram they actually show the hydrogen bonds, and you can see here the purine, the double ring, and the pyrimidine, the single ring. One other interesting thing about this diagram, which we'll talk about in more detail later, is that the artist has shown that on this side is drawing the DNA upside down. and that is to indicate that DNA actually has a direction, which is quite important when it comes to replication and protein synthesis. So DNA, these strands do have directions. Okay, that pentose sugar is called a ribose in DNA and RNA, and the difference, the reason that one's DNA, the letter D as opposed to the letter R, it has to do with the fact that in DNA the ribose is lacking in OH group. So no OH group, deoxy, and ribose the sugar, so deoxyribose. Okay, and it is an acid, so the full name for DNA, you might know this, deoxyribonucleic acid. And RNA is just ribonucleic acid. Okay, the third type of nucleic acid we'll look at briefly here is ATP, which stands for adenosine triphosphate. Now if you look at this carefully, this part of it looks very similar to what we were looking at before. This is the 5-sided sugar, and this looks just like the adenine. Okay, the difference is, instead of bonding to one phosphate, there's 2 phosphate groups attached here. So it's 3 phosphate groups attached, so it's triphosphate. Okay, there's a lot of misleading diagrams in some biology textbooks. It looks something like this. So this is not correct, but I'm going to show you this because it's so common. We know that ATP provides energy for the cell, and so sometimes you see it written like this. The ATP gets broken down into ADP, which is adenosine diphosphate, and an inorganic phosphate. So basically one of the phosphates breaks off, so it goes from being triphosphate to diphosphate, and here's the inorganic phosphate all by itself. And sometimes in textbooks they put looks like an explosion around the arrow here to make it look like the energy comes from this molecule breaking apart. Well, that's a little bit misleading because that's not actually what happens. Energy is not released like that when molecules break apart. Energy is released when smaller molecules bond together to make larger molecules. Right? Energy is released during synthesis reactions. So what actually happens is this inorganic phosphate, now that it's free, it can go off and it can bond with various other molecules in the cell. And when it does that, when it bonds with them, that's when the energy gets released. Okay? And after it's done that, then this inorganic phosphate may join up again with ADP, and this might go in the other direction to make ATP. So let's take a look at this diagram. This represents so energy from sunlight or from food. So this might be, the glucose that you had for your breakfast. What it's actually doing, that glucose is used to make ATP. What it's actually doing is it's taking the inorganic phosphate and it's bonding it back together with the ADP to make ATP again. Okay, so when we say ATP is being produced, what we're doing is we're basically attaching one of these phosphates back onto the ADP. Okay, so here's ATP, and then when your body requires energy, it breaks down again into ADP and inorganic phosphate. And then, like I was mentioning down here, this phosphate all by itself, this can go off and bond with other things temporarily, which is going to release energy and drive chemical reactions. And so on and so on. This cycle just keeps going back and forth in the mitochondria. So nearly all the cellular work depends on phosphorylating other compounds. Phosphorylating is a fancy way of saying adding phosphate. Yeah, that's what we were just talking about. And because this is a cycle, you could say that ATP is a renewable resource. And this is happening right now in the mitochondria of your cells. If you are using a lot of energy, so particularly in your muscle cells where you'd have a lot of mitochondria, scientists estimate that we consume about 10 million ATP molecules every second in a single cell. Okay, so that's breaking back down into ADP and inorganic phosphate, and then just like that, these join back together again to make ATP in cellular respiration.